All right, thanks, Gain. So I'm going to jump right into a little discussion, close that, of some of the work that's been done to organize data, some of these data that Gain just spoke about, for a variety of purposes. I want to thank my co-authors, and that really includes the whole 1KP consortium, as I'll say. I mean, this project really is a testament to the plant evolutionary biology community, and actually spelled large. Really, we had over 100 providers of tissues and RNAs, and it's been so important to the whole project. You know, this is all the contributors, the funding contributors. Actually, I should also mention that NSF has also contributed funding with bioinformatics, and also BGI and China National Gen Bank, providing a lot of the informatics tools that we've been using in the project. Okay, again, thanks so much to the providers. This is just a list of some of the folks that have been providing liverwort, just one group, and you know, we couldn't do this project without you, and I really hope that these resources will be useful to you and the whole plant science community. All right, so phylogenomics, two-sided coin, and with 1KP project, we've been trying to do both these things. On the one hand, use genome scale data or transcriptome scale data to reconstruct phylogenetic relationships. And we're really just scratching the surface in doing that of these data. We also want to then use this genome scale data to, you know, this phylogeny that's been developed to understand something about gain and loss of genes and genomes, do other genome scale types of comparisons. You know, enabled in part by the 1KP data and also the whole genome sequence data that hopefully is continuing to come back. So here's this basic idea of what 1KP has been doing with this transcriptomics project, Phylotranscriptomics. We've got a lot of materials, I said, from wonderful collaborators across the world. Extracted RNA, BGI did the sequencing, BGI assembled this using new software developed really in some ways for this project, but distributed widely. We then clustered those assemblies into gene families, our best estimate of what a gene family might be, so that we could make alignments and gene trees and species trees. So SOAP trans, I mentioned that there was this assembler that was developed for, you know, it was really motivated, development for analysis of 1KP data. It's been, you know, widely used now, and so that was a very nice product that, you know, spurred on by the 1KP project. As Gain said, if you want to look at most of these assemblies, you can go to this BLAST website and get it. And then, of course, data as it's being published is being put into Dryad, into iPlants data stores, and so on. Here's quite a little bit different look at the bioinformatics that we've been doing. In order to identify gene families, we've been relying on analyses of proteomes from fully sequenced genomes. Basically, what we've done is taken those genes from those proteomes and clustered them into gene families using MCL-based approaches, made HMMs from those so that we could use those HMMs to sort our transcriptome data into gene families and then do our phylogenetic analyses both on, you know, on individual gene families and then put it all together to analyze phylogenetic relations among organisms, using both coalescence-based approaches and supermatrix approaches. So this is basically the idea with this MCL-based approach to gene family classification or circumscription. We're taking genes from all these reference genomes, 31 in total, mostly angiosperms, but we have like 3 algae, Fisco, of course, and Salmonella. Do all against all blast, take this graph-based approach to identifying clusters based on those blast results, and then we end up with these circumscribed gene families and singletons as well, things that aren't starting gene families, and those might be interesting. We're looking into those singletons. One thing I will say, a widely used tool for doing this clustering is OrthoMCL or TribeMCL. A paper now that's just coming out in genome biology called OrthoFinder, Steve Kelly, Gain had mentioned, is done in his lab. And one thing I'll say about this is over here we see the kind of representation of the clustering that was done using OrthoMCL at various criteria, tightness of the for the clusters. And we see is that OrthoMCL is more inclusive. It's including more, making larger gene family clusters. And we think that those are, for a lot of our purposes, this is what we're going to, although not all. I mean, there are some instances where these gene families identified by OrthoMCL are a little too large. But for what we want to use for the phylogenomics to understand phylogenetic relationships are these single copy gene families. In that case, having this very loose circumscription of the gene families that are the most inclusive group of genes within the gene family, and still seeing that those are single copy across all of these 31 reference genomes, those something about them, in the face of gene and genome duplication occurring constantly, these seem to be persisting as single copy. Here's, so we had for this 31 gene set, we had 410 gene families that were only occurred once in 28 or 29 of the 31 genomes that we looked at. And here's a heat map showing how the presence and absence of those across our 1KP taxa. And we see that the coverage is actually really pretty good across our transcriptomes, with the exception of Chromista and Rhodophyta. Of course, we didn't have any genomes that we included in our circumscription for gene families of those, and of course they're outside of Viridiplantae. But still, we got enough genes here to use these as outgroups in our analyses. The chlorophytic algae are also a little thin in for some of these gene families, but still we've got a decent occupancy even for some of the chlorophytes. One thing I'll say about these single copy gene families, the one group of Go categories that is really showing overrepresentation in this set of single copy gene families are genes that are where the proteins are targeting the organelles, and actually within those things that have catalytic activity. So there's a bunch of Go terms across the various Go categorizations, and all of these kind of zero in on genes that are, or Go classifications that are having something to do with organellar metabolism. And here's the tree. You can't make a lot of this. I'm going to leave it to a lot of our other speakers to dig into this more deeply. But this gives you some idea of the relationships at the gross level and our sampling of things. So core eudicots are very well represented. This is blows up the core eudicots a little bit, but we knew we sampled a lot of angiosperms, a lot of core eudicots within those. But we've sampled across the Viridiplantae, and again, I said our outgroups are red and brown algae. So digging into some of these results, one of the things I want to just mention, because I don't think any of our speakers are going to be, what groups are most closely related to the land plants? And this is an earlier paper published last year in PNAS on the first 100 or so transcriptomes from 1KP, and we found that Zygmophyceae, I mean, this is a result that was found by others as well. We got much better support for it than previous studies. Zygmophyceae, rather than Chara or Coleochaetales, showed up as sister to land plants. And now with 1,021 taxa, we see that we have that same result. Here's Zygmophyceae. Here are the bryophytes. We have hornworts first, and then this clade, including mosses and liverworts, and then on up to the rest of the land plants. So those are the results. So you believe this tree? Should you take it at face value? Of course not. These are hypotheses. And what are some complications, some things that everyone should be very circumspect about with regard to these data? There's a lot of missing data. Okay, I showed the heat map. There's a lot of, it's not so bad, but there are a lot of missing data. Ortholog identification. We're taking this similarity-based approach to identifying and sorting, identifying gene sets and sorting our transcriptomes into those gene sets. Contamination, and we're taking steps. That's a very important thing. I've never seen a transcriptome data set that doesn't have some sort of contamination. And we've taken a number of steps, including blast-based approaches, trimming long branches to try to minimize those. Model misspecification. Of course, there's a lot of discussion going on about using supermatrix approaches versus coalescence-based approaches. And it is an issue even when we're talking about diversification over a billion years or 500 million to a billion years. So in this earlier paper that I mentioned, we noticed that however, when we took a coalescence-based approach, this result held up where the Zygmophyceae is sister to land plants. There were differences between the coalescence-based analysis and the concatenation analysis, and you can look at the paper to look at some of those. With our larger gene set, we're seeing that when we analyze the amino acids, that result holds up. The Zygmophyceae is sister, as a clade, is sister to land plants. However, when we analyze first and second codon positions and the nucleotides, we see that this terrestrial desmid ends up as sister to the rest of the land plants. An intriguing result. Basically, it's moving from the base of the Zygmophyceae over to the being sister to the land plants. So I suspect that this might be an artifact, and we're digging into that. But it's actually intriguing that this is a terrestrial desmid, and we should be talking about these as embryophytes, not land plants. There were land plants before there were embryophytes. All right, and I'll leave it there and be happy to take any questions.